Buenos días. Bienvenidos a este ciclo de seminarios cuyo título es Chemistry, an outstretched hand to society, la química, una mano tendida a la sociedad. Mi nombre es Francisco Ortega, soy el decano de la Facultad de Ciencias Químicas y únicamente quería darle las gracias a la vicedecana de Relaciones Internacionales y de Investigación, que es María Teresa Villalba, que a continuación va a tomar la palabra, por el empuje y la organización, digamos, de este seminario, así como a todas las personas que han participado. Dar las gracias a las 20 personas que van a participar. Este seminario es de alguna forma una continuación de uno que ya realizamos en la facultad el año pasado, que se titulaba ¿Qué investigan los jóvenes investigadores de la facultad? Solo que este año además hemos incorporado también a investigadores de otras facultades, de farmacia, de biológicas y de bellas artes que hacen su investigación, digamos, está dedicada a las ciencias químicas. Y quería agradecer a todas las personas que van a participar y singularmente en esta primera charla a Sara García Linares, que va a ser la que va a comenzar con el ciclo de estos seminarios. Y agradecerles a ustedes su presencia en el ciclo, siguiendo en este caso en esta primera charla. Y sin más le doy la palabra a Maite. Maite, cuando tú quieras.
Okay, thank you very much. Good morning, everyone, and welcome to this cycle of conferences in which, under the title Chemistry, a Hand Outstretched to Society, 20 talks related to chemistry, including biochemistry, will take place. 2 years ago, we organized an activity in which, through 30 short talks of about 15 minutes, we presented the research carried out at the Faculty of Chemistry. This activity allowed making visible some of our main research projects to the university community. 2 years later, and when our society and also our university are recovering of the negative consequences originated by the COVID-19 pandemic, we look forward resuming our activity with a new cycle of 20 talks, a similar philosophy, but adapted to the post-COVID era in several aspects. Firstly, since chemistry is not an exclusive prerogative of this faculty, but also there are excellent research groups at the Faculty of Pharmacy, Fine Arts, and Biology, we have extended the participation of this series of lectures to these faculties. Secondly, we have chosen the online format instead of face-to-face, taking advantage of the platform provided by the General Foundation of the UCM, to whom I want to thank the help and support to organize this activity. In addition, we thank to those who have contributed economically to this activity, the Spanish Biochemistry and Molecular Biology Society, the Royal Chemistry Society, Sectorial of Madrid, and Vice Director of Research and Transfer. This format will enable many more people to connect. Thirdly, the Spanish language is extremely rich in terms and expressions, but science is mainly written and discussed in English. Taking advantage of the fact that more and more researchers are joining our faculties after sometimes researching abroad, we have organized these talks in English. In this way, we will be able to broaden our audience to foreign institutions, some of which have been the hosts for these researchers. The lectures will take place on Thursday at 12:30 and will be announced every week, including the link of the registration form. Students who have enrolled for credit for this activity, please let me know by emailing at the address given in the announcement of this activity. Without further ado, I give the turn to the moderator of this talk, Professor Álvaro Martínez del Pozo, from the Department of Biochemistry and Molecular Biology, who will introduce the speaker and moderate the questions that you can write in the preguntas y respuestas panel, and you can vote these questions also. Professor Martinez, it's your turn.
Okay. Good morning, everybody. I am here to present to our speaker of today, Sara Garcia Linares. Sara joined our laboratory in 2009, still as an undergrad student. Simultaneously, she was also finishing her violin studies. 8 years later, in 2017, she defended her thesis, which was recognized by our Faculty of Chemistry with one of the extraordinary awards of that academic year. In between those dates, she also spent 6 months at the University of Abo Academy in Turku, Finland, where she joined the laboratory of Professor Peter Slote, a world leader in the field of the study of lipids, such as cholesterol and sphingomyelin. Once her PhD degree was obtained, Sara moved to Harvard for her postdoc. As a member of the highly exclusive program held between our Universidad Complutense and Harvard, she joined the laboratory of Professor Adrian Salik, where she spent 2 years. There, she worked in the characterization of GAS1, a very important protein for the correct development of human embryos, and the only protein known to be covalently modified by cholesterol. About 2 years ago, she came back home. Now she is in charge of our research group, specialized in the study of toxic proteins. She is meant to gradually become our boss in the short-term period. All these scientific tasks have been made compatible with her performance as a violinist in different orchestras, not only here in Móstoles, but also in Turku and Boston. In summary, Sara is now a highly mature but still young scientist and an accomplished violinist. She indeed possesses great communication skills, and you are about to confirm now. Therefore, Sara, now is your turn.
Thank you very much, Alvaro, and thank you, Maite, for having me in this cycle. Let me share my screen. You should be able to see the presentation now. Let me know if that's not the case. Okay, so good afternoon, everyone. My name is Sara. As Alvaro said, I'm a postdoctoral fellow at Complutense University of Madrid. I have the honor to launch this cycle, Chemistry, an Outstretched Hand to Society. And today I want to tell you about venoms. Are they enemies or allies? Well, survival is the main goal in nature. Animals and plants have developed different mechanisms in order to survive in nature. And some of them have strong paws and jaws to capture their prey. Others can run at incredible speed to escape from their predators. In our case, we have developed a complex brain that allows us to use the world around us for our benefit. For example, we are not especially fast, but we invented motor vehicles. So hunting for us is not a priority anymore. But what about animals that are tiny or animals that cannot move at all? How do they defend themselves from predators or capture their prey? Well, this is the case where venoms come in handy. Venoms have fascinated humankind throughout history, since the suicide of Cleopatra by the bite of an aspid, although, well, there are other versions that say she used some kind of poison or even that she was murdered, to the suicide of the philosopher Seneca. The history is full of stories about venoms. And nowadays, venoms are more in fashion than ever, because there are hundreds of research groups studying them, aiming to understand them, and also to be able to use them for our benefit. And there are even specialized scientific journals like this one that you're seeing here, Toxicon, is all about venoms. And I want you to look at this particular device because it's going to be a main character later in the talk. So As I said, venom field has grown exponentially. It's a huge thing right now. And actually last year, an European consortium was created to help organize and improve communication and collaboration between all the researchers working in the field. And this European initiative held its first Congress last week. And I was lucky to attend, and it is incredible how much research is being done in this field. Actually, our group was also invited to present some recent results of our own work, and I will talk about that a little bit later. There's no wonder this field is so vibrant with so many researchers in it, because there are thousands of venomous species on Earth: snakes, scorpions, centipedes, frogs, you name it. And they are the source of incredible biological compounds with a great potential. And here you have my favorite venomous animal, a sea anemone. And I say it's my favorite because I have been studying the venom of sea anemones for more than 10 years. My doctoral thesis was about one of the components of this venom. And sea anemones are animals that live attached to rocks or to the bottom of the ocean by their petal disc. The oral disc is where they intake their prey to be digested. They mainly feed on small fish and small crustaceans. So how these prey end up in the oral disc? They don't go willingly. There's another part in the sea anemones, which are the tentacles, and in the tentacles we can find the stinging cells. It's a specialized type of cell called cnidocyte. And inside the cnidocyte, there's this organelle called nematocyst, which is the one that contains the venom. So anytime a prey or a human comes close enough to the anemone's tentacles to touch this trigger here called cnidocyl, then the nematocyst ejects from the cell and deploys like a spear. You pull the trigger and the spear comes out. And we are going to see this process in motion with this wonderful animation by Ted about jellyfish. Jellyfish are related to sea anemones, and actually they have the same type of stinging cells. So let's watch the animation.
Venom is ejected via a nematocyst, a whip-like hollow tubule which lies coiled under high osmotic pressure. When mechanical or chemical stimuli activate an external trigger, the lid of the cell pops open and seawater rushes in. This forces a microscopic barbed harpoon to shoot out, penetrate, and inject venom into its victim.
Yeah, not very nice. If you have been stung by a jellyfish sometime, you know. So the nematocyst deployment is one of the fastest biochemical processes in nature. And in this slow motion video, we are going to see how fast this happens. So here you're going to see the beginning of the deployment marked with a green T. And after the ending of the deployment, only a few milliseconds go by. So like I said, very fast. And in the next shot of the video, you're going to see how after deployment of the tubule, the venom is clearly delivered through the nematocyst. So what's this sea anemone venom? What's inside this venom? So sea anemone's venom is mainly composed of different toxic polypeptides or small proteins of around 200 amino acids. So there are different kinds like neurotoxins, which are toxins that affect neurons, as their name indicates, and they are responsible for the pain that we feel when we touch one of these animals. It has also phospholipases that degrade the phospholipids in our cell membranes, and cytolysins. Cytolysins are molecules that make holes in the cell membranes, and they are responsible for the inflammation caused by the sting of these animals. Inside this last group of proteins, the cytolysins, we find a family of proteins called actinoporines. Actinoporines are produced by many sea anemones, and so far they have been detected in at least 20 different sea anemone species, but only 4 of those actinoporines have been extensively studied during the last decades. We have echinatoxin 2 from Actinia echina. It's an anemone that we can find in the Mediterranean Sea. We have sticolysins 1 and 2 from Sticodactyla lianthus. This anemone is from the Caribbean Sea. And Fragacia toxin C from Actinia fragacia, an anemone that we can find all the way from Norway to Africa. And it is called also the strawberry anemone, because you can see that it has the looks of an strawberry. So if we want to study venoms or we want to use them for any purpose, the first thing we need to do is obtain them. And the most obvious way to do that is to turn to the biological source, which is the venomous animals themselves. And you can see here someone milking the venom of a snake. It's a picture I guess we all have seen at some point. Less common may be this electrical stimulation of a centipede to force it to release the venom. It's a very low electrical current, so no damage done. And you can also extract venom from the venomous glands of spiders, scorpions, also from the tentacles of sea anemones. But the natural sources have some drawbacks. For example, you first need to find these animals in nature, which can be hard or time-consuming. You also have to handle them, and in some cases, I guess that's a little bit dangerous. And last but not least, the amount of venom that you can get from these animals, sometimes it's too small to do research or to do anything with them. Imagine, for example, a black widow spider, it's like super tiny, and the amount of venom that you can get from it is ridiculous. So sometimes there's no other option and you have to deal with this inconvenience. But if you're lucky, you can turn to an alternative, which is genetic engineering. And for those of you who are less familiarized with this concept, let's go into review the basics of it. So here we have a bacterium. Bacteria are single-cell living organisms, and as we do, they have chromosomal DNA, but they also have plasmids. Plasmids are small DNA molecules, normally circular, and they are external of the chromosomal DNA. They can replicate independently. And as DNA does the job, plasmids do whatever DNA does. And what does DNA do? Well, let me introduce you the central dogma of molecular biology. And it goes like this. In the nucleus of the cell, we have the DNA. And the DNA can replicate to make a copy of itself. And then it gets transcribed to mRNA or messenger RNA. And we have heard a lot about of this molecule this last year because it is the key component of some of coronavirus's vaccines like Pfizer. This mRNA is going to leave the nucleus of the cell and it's going to get translated into proteins. And proteins are molecules that make basically everything in a living organism. So we can use genetic engineering this way. We have the genetic information of a protein that we want to produce, and it can be even a human protein, for example. And we can take this information and put it inside a bacterial plasmid, making what we call a recombinant plasmid, which will have the genetic information of our protein and the genetic information necessary to function inside the bacteria. Once we have the recombinant plasmid, we put it in a bacteria culture and we heat. And this treatment allows the plasmid to go inside the bacteria, but not all of them. Only a few of them will take the plasmid in. Most of them will be non-transformed. And we don't want this non-transformed bacteria for anything because only this one will be able to produce our protein. So how do we get rid of non-transformed bacteria? Well, the recombinant plasmids have other information, another information that allows us to select the transformed bacteria. It has an antibiotic resistance gene, which makes the bacteria that take the plasmid resistant to that particular antibiotic. So we have the culture with the transformed and non-transformed bacteria, and we can put it in a plate in the presence of that antibiotic. So only the transformed bacteria will survive and the other ones will die. So we can take one of these surviving colonies, put it in a bigger culture and grow a big number of bacteria. And then we trigger the expression of the plasmid. So our gene will get transcribed into mRNA, and this mRNA will get translated into the protein that we want to produce. So just to sum up, we have the recombinant plasmid with the genetic information of the protein and the bacterial plasmid. We put it in a culture of bacteria, we heat. Some of them will get transformed. The other ones will die once we put the culture in the presence of an antibiotic. Only the transformed ones will survive. We grow them in a big number, and then we trigger the expression of the plasmid to get the protein. And the advantage of this is because it is relatively easy to grow microorganisms like bacteria or yeast, we can get a big amount of protein necessary to do research or to use it for any other purpose. And I say any other purpose because this process, it's used in many different ways. Let me put you an example. Before molecular biology was developed, insulin to treat diabetic patients was obtained from pig pancreas. And here you have a picture of 8 tons of pig pancreas needed to produce 250 milliliters of insulin. Luckily today, it is produced safely with transgenic yeasts. Okay, going back to sea anemones and the actinoporines, how these proteins work. So as I said, they are cytolysins. They are proteins that make holes in a lipid membrane. So they find a lipid membrane, they attach to it, they extend this part of the protein and insert it into the membrane. And then several of these proteins come together, and with the helices inserted in the membrane, they make the hole. So what happens to a cell if you make a hole in its membrane? Well, nothing good. So here we have an erythrocyte, to use it as an example, and they are actually widely used in the pore-forming proteins research. So the erythrocyte is happy because the cell membrane is thought to isolate a cell from the outside media. So when we add our actinoporine and we make the pores in the membrane, we break that barrier, then things that should stay inside go outside, and things that should stay outside go inside. And all this creates an imbalance that ultimately kills the cell. And in our case, for example, this causes some of the symptoms that we experienced when we touch one of these animals. So how do we study venoms in the lab? So I'm going to tell you about experiments related to pore-forming proteins, but remember that venoms are composed of different things, and there are different approaches to study them. So for example, I said erythrocytes are widely used in this field, and hemolysis experiment is widely used to test the activity of a pore-forming protein. Here's a real experiment that I did many years ago, although we are still doing it routinely. And erythrocytes are big enough to scatter visible light. And that means that a red cell blood suspension is turbid. You cannot see through it. But when we add our actinoporine in a concentration increasing manner, we see how the erythrocytes die until we get to the highest concentration. All the erythrocytes are dead. And now the suspension is not turbid anymore. It's completely clear and you can see through it. We can use dedicated instruments for this to measure how transparent our suspension is, and we can relate the transparency with the percentage of hemolysis. The more transparent it is, the more hemolysis we are seeing. So we can represent the percentage of hemolysis in function of the protein concentration, and we get a curve like the one I'm showing you. And from this curve, we can get this value, HC50, which is the protein concentration needed to kill half of the erythrocytes that we have. And this value will tell us if an actinoporine or a pore-forming protein is good or bad at doing pores, and compare one to another. We have another experiment to test pore-forming activity, which is releasing the content of a lipid vesicle. In this case, this vesicle contains calcine, and calcine is a fluorescent molecule. But when it is inside the lipid vesicle, the fluorescence emission is really, really low. So if we add our actinoporine, it makes a hole in the membrane, the calcine escapes to the outside, and fluorescence emission increases like you are seeing in blue. Both hemolysis and calcine release experiments allow us to determine the particular role of specific residues in the protein. For example, if we change this amino acid here and suddenly we see that the protein is really bad at doing pores, we know that that particular residue was really important for protein function. And calcine release experiment gives yet another advantage, and it is that we make this lipid vesicle, so we can choose lipid composition. In that way, we can study not only the role of protein amino acids in the function, but also to determine the role of the lipids in the pore-forming activity. Okay, I'm going to tell you about another experiment that we do, and this one allows us to actually see the pore. And this is not trivial, because keep in mind that we are talking about a structure of only a few nanometers. So we can use nanodiscs for that. And nanodiscs are like little pieces of lipid membranes, and we can add our actinoporine, reconstitute the pore inside the nanodisc, and then use an electron microscope to take pictures of it. And this is a very elaborate technique in terms of sample preparation, image acquisition, and also processing of the data. And here are some real pictures of our own pore made in collaboration with a group from the National Center for Biotechnology here in Madrid. Okay, so why do we study venoms? Well, the obvious reason is to formulate anti-venoms against species that are particularly dangerous to humans. According to the World Health Organization, there's an estimate of 5.4 million people bitten each year by snakes, with up to 2.7 million envenomings. Around 1,000 to 138,000 people die each year because of snake bites, and around 3 times as many amputations and other permanent disabilities are caused by snake bites annually. Bites by venomous snakes cause many symptoms like paralysis that may prevent breathing, bleeding disorders that can lead to a fatal hemorrhage, irreversible kidney failure, and tissue damage that can cause permanent disability and limb amputation. Agricultural workers and children are the most affected. Children often suffer more severe effects than adults because their bodies are smaller and the venom has more potency in them. So, for example, the Indian corporate genomic information was studied a few years ago, well, last year actually, and they found 19 venom-specific toxins in its genome, which included neurotoxins, cytolysins, and much more components that cause many symptoms like muscle tissue damage, paralysis, hemorrhage, cardiac arrest, breathing failure, many bad things. They also found accessory venom proteins that cooperated with the venom components. Well, what we can do with this genetic information, with this genomic information, is to study it and indeed identify in particular components of the venom. And these particular components, we can use them to apply special protocols to produce specific antibodies against these components. And we can use those antibodies as anti-venoms to treat these envenomations. Years ago, for example, researchers found an antidote for the most lethal creature on Earth, which is the Australian box jellyfish. The sting of this animal can cause necrosis of the skin, excruciating pain, and if the dose of the venom is high enough, even death within minutes by cardiac arrest. And the venom of one individual can kill 600 people. Well, what the researchers did was to use CRISPR genome editing techniques to create this antidote that blocks the symptoms within 15 minutes after application. CRISPR technique, maybe you have heard of it because it earned its inventors the Nobel Prize in Chemistry in 2020. And it's an invention that actually started in Alicante, Spain. And if you haven't heard of the technique, I encourage you to read about it because it is amazing what we can do with it nowadays. Okay, but we can go further in the study of venoms and go from merely studying them to combat them to actually use them in our benefit. And there are many studies around the world aiming at using venoms to kill cancer cells. And I'm going to show you some examples. So we have a peptide from the venom of an octopus that is being used to kill melanoma cells. This is a work from a group in Madrid. The venom of jellyfish showed to stop cell cycle in hepatocellular carcinoma cells. The venom of the Jararaca snake modulates cancer-related proteins in breast tumor cells. And melittin, which is a key component in bee venom, it showed to suppress cancer-related processes in human gastric cancer cells. And these are just a few examples of what it's been doing out there, but please keep in mind that I'm talking about cells in a culture plate. I am not talking about treatment of living organisms. This would be a first step, and it may or may not translate into future treatments. We can use venoms also in other areas different than health. Plagues, for example, are a very big issue because they spoil harvests, causing millions of dollars losses. And taking into account that we are already around 8 billion people in the world and growing, and we need to feed all those people, losing crops is a very alarming problem. And commonly used pesticides can have harmful effect on the environment, on the health of humans, or beneficial species like bees. So in our group, apart from the Anemones venoms, we are studying performing proteins from the venom of the black widow spider, which prey is actually insects. So these proteins would be a perfect natural biopesticide with the added advantage of being innocuous to humans and also biodegradable. Let me tell you about a new exciting project that we have in the lab. The goal of this project is to change the actinoporine pore to confer it with catalytic activity. So in normal conditions, actinoporines make a pore that is inert. Nothing happens, just it's a hole and things go from one side to another. But a group in Barcelona we collaborate with, they suggested few modifications that we could do in the pore to confer it activity, catalytic activity. So we made those proteins, we put them in nanodisc, and we tried it to with a battery of chemical substances. And actually, some of them showed to be modified when they pass through the pore. And this is an amazing result just as it is. Like we took a protein that did nothing and we changed it, and now there's an activity that it wasn't there before. But the result is even more exciting because some of these products that were modified when passing through the pore, they were plastic-like compounds. This means that in the future, we may use this system to degrade plastics waste, which is a huge breakthrough in environmental. Okay, so we have talked about venoms to formulate antivenoms, also to kill cancer cells, and our hypothesis that they could be used as biopesticides or as biological nanoreactors. But let me tell you about an example of something that it is actually in the market. So this is a cone snail, and of course, it is a venomous animal, and it feeds on small fish and small crustaceans, and it does that by injecting a venom with this harpoon here. And we are also going to watch this in action because this is one of my favorite National Geographic videos.
Extendable proboscis is packing a concealed weapon, a tiny hollow harpoon made of chitin, the same tough stuff in a lobster shell. And the end of the proboscis is tricked out with receptors, taste buds, that help it close in on its target. When it strikes, the snail's pace jumps to light speed. The embedded harpoon doubles as a hypodermic needle to inject the victim with paralyzing venom.
So one of the components of the cone snail venom is a neurotoxin that has been used to formulate a drug against chronic pain. Siconopite, which is the name, is the first pain reliever from a cone snail toxin venom. And it was approved by the FDA in 2004 and by the European Commission in 2005. And it works by blocking neural transmission. So this drug has two great advantages. It's like 1,000 times more potent than morphine, but it doesn't cause addiction. And as you know, opioid addiction is a very big problem in the US. But there are also important drawbacks with this drug. It has to be administered directly into the spinal cord fluid. And in some cases, it shows very severe side effects. So it is only prescribed in severe cases of chronic pain. But it is the perfect example of how we can actually use venoms in our benefit. So I hope I convinced you that venomous animals, although scary and dangerous, they are the source of amazing biological compounds that can be used. They have great potential if we know how to take advantage of them. Well, thank you very much for your attention. I hope you have enjoyed the talk. Tune every Thursday at 12:30 for many interesting talks, and I'll be happy to answer any questions.
Okay, Sarah, nice talk.
Thank you.
I hope everybody has enjoyed as much as I did. No questions so far, so I am going to begin. I am going to begin with a question. many of these pore-forming toxins have not the usual protein receptor, but a lipid receptor. And in many cases, this lipid molecule is sphingomyelin and most especially cholesterol. And can you explain to the audience why cholesterol is the target that many microorganisms and venomous invertebrates have chosen as target? Could you explain that?
Yeah, that's a great question. Well, I guess many of you have seen Finding Nemo. So you remember that actually this fish lives in an anemone, in a sea anemone. Like, why the anemone that feed on fish doesn't attack or affect this fish? So actually invertebrates and other animals have different kind of lipids in their membranes. So for example, higher vertebrates like humans, we have cholesterol in our membranes. So that's why pore-forming proteins affect to us, but not other animals.
So it's a safe insurance, no?
Yeah.
Another question, so another suggestion, if you could explain, you know, in our group, as you know very well, we're using immunotoxins. I wonder if you could elaborate a little bit about how toxins and antibodies can help us to fight against some diseases.
Yeah, so immunotoxins, they are a chimeric molecule with a toxin linked to an antibody that basically recognizes something that is particular to certain type of cancer cells. So in that way, we can take this toxin to a specific cancer cell, and it would be only effective on these cancer cells and not the healthy ones, avoiding all the side effects that, for example, chemotherapy has on patients because it kills also healthy cells.
Okay, no questions yet.
Sorry, Álvaro, sorry that I interrupt, but I think that there are several questions in the chat panel. I don't know, because maybe
let me ask a question that Begoña has put now in the questions, and then I'll look at the chat. Okay. So, Sarah, Begoña asked that it is, if she understood well, venoms are usually produced in yeast. Her question is, do you use other systems or other type of cells in those cases that you need to keep post-translational modifications that yeast cannot achieve? Or do you artificially modify them in the lab? And let me add something. Why do we use some yeast and no others?
Yeah, so actually what I use in the lab is bacteria, because we are lucky enough that our proteins are very simple structurally, so we can use bacteria because there is nothing complicated to do with the proteins afterwards. But some proteins need to be modified after they are translated, and bacteria cannot make some of these modifications. So in that case, we go to yeast, which is still a single cell living organism, but it is eukaryotic, so it can do more things than bacteria. And we usually use yeast or bacteria that are generally regarded as safe. Like, we know exactly all the genomic information, and we know how to modify them to use them in heterologous production of protein.
Thanks, Sarah. Rian Sares asked you about the size of the pore.
Yeah, it's like a few nanometers. I think it's it's like 6 nanometers on the upper part and 2 nanometers the down part.
Okay, actually, I didn't understand well the question, because the question was originally from Lucia, and she's asking if it would be possible to create larger, I guess she means wider or narrower.
Oh yeah, sure. I mean, I didn't want to go deeper into actinoporines because we were talking about venoms in general, but pore-forming proteins are a huge family, and there are different kinds. And actinoporines, for example, are alpha pore-forming proteins because the helices are the ones that insert into the membrane. But there are beta pore-forming toxins, and those ones can be, the pore can be made of like dozens of monomers. So the pore in that case is huge.
Okay. Thank you very much. Laura Nogues is asking if the mechanisms by which venoms kills cancer cells is specific of each venom and cancer subtype, or if there is any general mechanism common to all of them.
In that case, I don't know if I have a clear answer, because like actually, well, many of these studies have a problem with selectivity. So in these kind of studies, many healthy cells also die by the toxin. So there's a little specificity for the toxins, but not too much. So that's why, for example, as Alvaro said, immunotoxins could help with that problem, because it would actually take the toxin to the right cells to kill.
That's the approach we're using to circumvent that inconvenience. Okay.
Mm-hmm.
There's another one, Adriana Espada. This is a personal question, let's say. How did you discover that venoms, natural venoms, would be your subject of research, and why have you dedicated so much time to this study? Why did you choose this subject?
Actually, I think I was taken by the subject. Like, I first chose the group to work with, and because they were working on this project, I just immersed so much in it that I just thought that I would love to keep exploring the field, because it is actually amazing. And after 12 years in the lab, we still get new results and new possible applications to these proteins. So I guess it was luck.
Rian Sares Muñoz is asking if we know, or if you know something about neurodegenerative diseases. Do you have any hint about how venom application can be used in this field?
I don't know if there's something on that. I will have to check.
Probably there's some, especially regarding, as you mentioned, probably regarding pain, mostly. So
yeah.
So far. Javier Garcia Virceda, he is asking if you have thought if the venoms could be an application against COVID, or maybe this is so new.
Against COVID. Now everything is about COVID. I don't know if we could use venoms to, like maybe if we find a particular aspect of the lipid membrane of the virus that makes it different to other cells, we could formulate a protein venom or a component of a venom that could target specifically that component of the virus without affecting other cells. I guess the lipid membrane would be the key, or maybe, well, a protein receptor, but virus has so few proteins that I would go with the lipids.
Edna Nebreda is asking you if, regarding antivenoms, do antivenoms based on antibodies work directly by opsonization of the peptides, or do they rely on the activation of an immune response to eliminate them? And she thanks you for the presentation.
Yeah, I'm not sure, but I guess they have to be really fast. So my guess is, and because how they are formulated, like using the actual component of the venom, I will go with the opsonization of the antigen.
I think so too.
Yeah.
Most of the antivenoms are antibodies.
Yeah.
So no, let me see. I'm new with this. This is a very specific question from Eva Alvarez. She says, I wanted to ask if nowadays it is known about how the CRISPR technique can be applied in the cure of Australian box jellyfish.
Yeah, that was done 2 years ago. I haven't read the actual article, but they use CRISPR to formulate the antidote.
Okay. I didn't know that. Any other questions?
Can I ask a question?
Sure.
Yeah, of course you can.
I want to know how many toxins different toxins molecules can have one anemone, for instance, or a spider, and if they have different functions in terms of their toxicity, not their function of the.
Yeah. So actinoporines, for example, are cytolysins. They make holes. That's their job. But neurotoxins, for example, they usually target ion channels in our cell membranes. Like they mess up our electrical current in the membranes that it's needed for the function of neurons. And phospholipases, as I said, they degrade the phospholipids in our cell membranes. So yeah, the venom is a complex battery of different things going to different sites in the cell and different functions targeted to, like, basically, for example, the poor fish eaten by the cone snail, it was totally paralyzed because of the neurotoxins.
Can I add something?
Yeah.
They actually, you have a race between the target and the predators. Yeah. So venoms co-evolve with the immune system of the animals, or the animals they target. So one of the advantages and also the disadvantages of using venoms is that most of them are highly specific. So the disadvantage is the application is less general, but once you have a good application, it's specific, no side effects, theoretically. Well, you have many congratulations in the chat, but I guess you see those. Well,
I haven't opened it yet.
Okay, there's another question by Adrián Espada. He's asking if you know of some mammal who has the ability to have a venom to survive.
What was the question? Like they beaten and survive?
He's asking about mammals with venom that produce venom.
Mammals, yeah, I'm sure there are.
Platypus.
Which one is that?
Platypus. Platypus has a ornitorrinco.
Oh yeah. Okay. Yeah. I knew. Yeah. I knew there was some mammals with venoms.
Very strange mammal.
It lays eggs.
Maybe another one. Maybe some other one.
Yeah.
But you can also elaborate on the basis of this question how mammals and even the immune system use a very similar approach to kill pathogens who invade our bloodstream, as actinoporines, as pore-forming proteins.
Mm-hmm. Yeah, I mean like natural killing cells, you mean?
Or pore-forming proteins. What I mean is pore-forming proteins from our own immune system.
Oh, like the complex, the MAP complex. Yeah. It's one of the tools that our immune system has, like making holes in the membranes of the pathogen to kill it.
It's not a venom, strictly speaking, but it's the same approach and mechanism.
Yeah, it's the same mechanism.
Jose Santos asks if antivenoms can be studied through the biology of the animal producing it. I don't understand very well the question. I don't know. If antivenoms can be studied through the biology of the animal producing it.
Yeah, well, I'm going to answer what I think he's asking. Like the Cobra case, like you take the whole genome, and you can, from the sequence, you can see if there are different products of this venom. And I guess you can do two different things. You can choose what component you're going to target with the antibody, or you can take a few of them and make a more general antibody that is less specific but wider in action. I don't know if that was the question.
I think that could be the question. He's explaining that maybe the animal producing it could be immune to it.
Oh, okay. So like finding inside that same animal something that can act as an anti-venom. Yeah, but I'm not sure if the animal is immune. I think there are other mechanisms that keep the animal immune, not something like physical. Like, for example, the anemone case, the fish is immune because it lacks something that the venom needs to function. So I think that the animal that produced the venom is immune to it, but because it hasn't the right components for the venom to act, not because it has something that nulls the venom.
And also compartmentation.
Yeah.
Okay, maybe we can finish the talk. This talk. I thanks a lot, Sarah, because it was a nice talk.
Thank you.
And thanks a lot to be the first also.
It was a great pressure.
I know. But I want only to say to our audience that next talk will be Thursday, September 38. José Santos Barahona is a professor contratado doctor of the Organic Chemistry Department, and we'll talk about on surface synthesis of novel polymers. I am sure that it's going to be a very, very nice talk too. Thus, I hope we have the next talk was so exciting as this one, and then see you next Thursday, everybody.